Now, in bacterial photosynthesis, a single photosystem is involved instead of two like in green plants and algae. So when an electron is energized by an absorption of light, it's ejected from the photosystem's reaction center. So let's take a look at that. There's our photon now ejecting our electron. And remember, this bumps that electron up to the next level. So the excited electron then passes to the ferredoxin. So here's the bacterial electron transport chain. So there's our electron passing to ferredoxin now. Okay? Just like similar to plants and green algae. Now, the excited electron then goes to ferredoxin, then to the B6F complex. So there's another similarity. Ferredoxin, B6F complex, and ADP now reunited to form ATP. Now, the energy released during this electron transport is used to produce ATP. Since the excited electron returns to the original reaction center, like you see here, it's called cyclic photophosphorylation. So, let's follow this electron again. So, remember, in a cycle now, we return back to the beginning and start over again with the same things, like the water cycle. So, here we see our excited electron, our photon, hitting our electron, then ferredoxin, ADP, the B6F complex pumps that hydrogen and reunites ADP with phosphate. So again, you get that phosphorylation just like you do with two reaction centers. And then plastocyanin now, which is now an electron carrier in the electron transport chain, passes that electron back to the reaction center. Now remember, in non-cyclic photophosphorylation, electrons are replaced completely differently, right? So the photosystem 2 reaction center electrons replace the photosystem 1 electrons, and then the photosystem 2 electrons are replaced by what? Right, the stripped electrons from the water molecule that's been split. Now, this is completely different in bacteria. So again, that is absolutely a question somewhere on the AP exam. So you got to know the difference between cyclic and non-cyclic photophosphorylation, and you got to know the organisms that it occurs in. Cyclic now is bacteria that we've seen here. Non-cyclic is algae and green plants. So make sure you get that clearly down in your notes. It's very important for you to understand. Okay, now just a quick review of non-cyclic photophosphorylation before we get to the next phase of photosynthesis in plants and green algae, called the Calvin cycle. Now, it's worthwhile to remember the purpose of photosynthesis, which is to generate and to make organic molecule, or generate ATP, and to make organic molecules like carbohydrates, right? Now, a question for you. Which of these functions have we accomplished so far? Now, if you said energy production, you'd be correct, in the form of ATP, right? So write in your notes that the two molecules generated from the light-dependent reactions, sometimes referred to as the electron transport chain, would be ATP and NADPH. Also write that it's these two molecules that will be needed and used in the next phase of photosynthesis called the Calvin cycle. Now, a quick rundown of the electron transport chain, also called the light reactions, also called the light-dependent reactions, just to get it right in your mind. Now, photosystem 2s, also called P680s, if you don't have that in your notes, scribble it down now. Photosystem 2 is referred to as P680 because this chlorophyll and these electrons are sensitive to light at 680 nanometers. Now, let's quickly write down that photosystem 2 is referred to as PS7 or P700 for the same reasons. This reaction center is sensitive to light at 700 nanometers. When we go to our lab in Lake Superior State University, we're going to have to calibrate our spec 20s to the light frequency that our photosystem is sensitive to. We're going to use photosystem 2 of some spinach plant chloroplasts. So we're going to set that spec 20 at 680 nanometers. Now, so those P680 pigments absorb photons of light, which generate excited electrons that will enter this electron transport chain. So again, here's our photons now exciting these electrons, and those electrons then are stripped or are passed along this electron transport chain. Now, and at the same time, right down here now, we have our water splitting enzyme, all right, that splits water, and its electrons are used to replace those lost in this first step. The two O's, remember, it's two water molecules that are split. That might be something to re-emphasize in your notes. It's two water molecules that are split. So that makes sense because here's one oxygen, but there's another oxygen from the other water molecule. So those two combine to form this breathable oxygen that's released off into the air. Now, we're using these hydrogen ions now called protons in this whole process eventually of making ATP. So three things water gives us. Electrons replace now these photosystem 2 electrons. Two oxygens are united to form oxygen, O2, that's given off into the atmosphere. And these hydrogen ions now start to fill up this thylakoid space and are going to be used to form ATP. Now let's review how ATP is formed in this first step. Remember that excited electrons pass from chlorophyll to the primary electron acceptor plastoquinone, which is right here. Then electrons are passed down this electron transport chain to the B6F complex, also called the cytochrome complex. Write that down in your notes. This is our B6F right here. So plastoquinone to B6F, which is sometimes called the cytochrome complex. So get that in your notes. Now, this B6F complex is also referred to as a proton pump. All right? So, three things. B6F, so this is your B6F, cytochrome complex, or proton pump. You could see any of those on the exam, so you got to be familiar with what's going on. All right, then each time an electron is passed, it releases a little bit of energy which is used to pump protons from the stroma. Here's our stroma right here, see it? From our stroma into the thylakoid space. So we're pumping protons in here now, as well as getting them from the water molecules. So now this thylakoid space is becoming concentrated with hydrogen ions. Now, what happens now is those protons then diffuse back into the stroma and pass through the enzyme ATP synthase. So here's our ATP synthase. It should be right up here, but it's not. They've got it separated so we can talk about it. So those protons now diffuse out. Why? Because the electron or the concentration is greater in the thylakoid space, and those protons now are used to reunite or phosphorylize ADP into ATP. And this ATP is then going to be used for the Calvin cycle, which is the next phase, or the dark reactions they call them, which is not necessarily correct. We'll talk about that a little bit more here in a bit. Okay, then ATP synthase unites ADP with a phosphate, allowing it to make ATP. We call that phosphorylation, and then that enters the Calvin cycle. Okay, now let's review how NADPH is formed. Now, photosystem 1's, also remember that photosystem 1's called P700 because it's sensitive to light at 700 nanometers, which is the wavelength. That its chlorophylls absorb light and pass excited electrons to the primary electron acceptor ferredoxin, which is right here. So instead of plastoquinone, all right, the second phase of the light reactions, ferredoxin now is the electron acceptor. Now, photosystem 1's electrons are replaced by the electrons passed from the electron transport chain of photosystem 2. So those electrons are passed along, and now they replace these electrons that are lost from this photon of light bumping these photosystem electrons. So they're replaced right here. So photosystem 1's electrons then pass down to the carrier molecule NADP reductase, right here, which unites NADP with a hydrogen ion. Okay, we know there's hydrogen ions out here. They're reunited with a hydrogen ion to form NADPH. Then NADPH is sent to the Calvin cycle in the stroma. So two things now are formed in this first phase is light-dependent reactions. Also will be used in the Calvin cycle. NADPH and ATP. All right. Okay. Now that we know how ATP and NADPH are generated in the light-dependent reactions and passed on to the Calvin cycle, let's take a crack at understanding what happens and where it happens in this important second set of reactions. Excuse me. Okay, first of all, write that the Calvin cycle is a series of reactions that take place in the stroma of the chloroplast, not the thylakoids, like you see here in this diagram. Okay? It occurs in the stroma, not in the thylakoids. Now, also write that these reactions result in the conversion of carbon dioxide into the organic molecules needed to build new cells, new cell parts and cells, and carbohydrates. Also add that the Calvin cycle requires the products of the light reactions to drive the synthesis of these organic molecules. Again, synthesis just simply means to build, is all it means. All right, take a deep breath. Let's try and make some sense of this process. Now remember, it's really important to remember the difference in the goals between the light-dependent reactions and the Calvin cycle, sometimes referred to as the dark reactions. All right, the goal of the Calvin cycle is to generate energy. We were always generating ATP and organic molecules. And that plants take in carbon dioxide through the stoma in their leaves, which is kind of like the pores in your skin. So plants take in this carbon dioxide through the stoma. Okay, so write in your notes that the exchange of oxygen and carbon dioxide happens in the leaf, as well as the loss of water vapor and transpiration that happens in the leaf too, through pores called the stoma. Stomata is actually the plural. Singular is stoma. And that normally stomata open when the light strikes the leaf in the morning. And then they close during the night. Also write that the pores are opened and closed by guard cells. You see the guard cells right here. And you also see the guard cells with chloroplasts in them. Now, they actually contain the chloroplast. So this is the source of carbon dioxide that the plant will use in the Calvin cycle. And it's worth looking at the chemical equation for photosynthesis and making some sense out of it now that you know about the light-dependent reactions. So the reactants are water and carbon dioxide, so here they are, water and carbon dioxide. We now know how important the water is to this process, right? Water split by the water splitting enzyme, and its electrons are used for the electron transport chain. Its oxygen, both oxygens reunite to form breathable oxygen, you see here, okay? And then its hydrogen ions are used to make ATP and NADPH. Now, carbon dioxide we see here. This is the molecule that will be used to make organic molecules you see here on the right. It makes sense that the carbon and oxygen now will be the building blocks of the sugars and carbohydrates here on the right. Okay, so when entering the Calvin cycle now, and get this in your notes, are three substances. Can you list them? If you said ATP, NADPH, and CO2, you'd be correct. Okay, that's it for part 3. Take a picture of those notes, submit them to Moodle, grab the study guide, answer the questions, and I'll see you back here for a more in-depth discussion of the Calvin cycle in part 4. Bye.